using whole genome sequencer data. So without delay, I'd like to let you know how to design. To design, initially we need to have a DNA sequence. So for getting a DNA sequence, we need to go through the National Library of Medicine. It's NCBI, so that we can easily get our DNA sequence. So I have taken the sequence of a bifidal bacterium uh, whole genome sequencer data, nucleotide sequence. So using this sequence, I'm going to design a primer with the Prime 3 software. So to design, initially I will select the sequence which I have already copied, and afterwards I put this sequence inside the Primer 3 website database. On doing paste, I need to have like primer without accession number and other text. So I have omitted all the extra informations like a text and accession number, and it is necessary to have like a forward and reverse primer. To get a forward and reverse primer, we have to pick the left and right primer, which is already tick here. So now I have to pick primer, and also we have different options. We can look it on those options too, and we can set according to our desire. But today, with this video, I'm going to let you know about the automated primer construction process. So I will go for the primer, and with this software I have a result. So I have left primer with the starting with the 11 nucleotides, and the length is 20 nucleotide sequence, and its GC percentage is 55, and 3 prime here pin sequence 0, of which we have to omit, which is already mentioned here. And here is given the sequence starting of the primer, like it is started with CATCAG, like that. This is the forward primer, CATCAG, you can see here, and this is represented by arrow. Like for the reverse primer, which is of like 11 nucleotide sequence. Yes, this is 11 nucleotide sequence. This is the left. So this primer has this kinds of area, you can see there. Okay, in this way you can design automated primer of DNA to identify the specific gene in the DNA sequence. And you have a multiple option of primer for this selection, having almost 20 sequence of each forward and length sequence of 20 base pair, and with this different nucleotide sequence. So this is the easiest way to design primer, and you can use this primer for the PCR amplification of a DNA sequence of a gene, so that you can identify a particular sequence of a gene with this forward and reverse primers, and you can have multiple copies of gene amplifying the specific region of a DNA. Thank you so much for your time and attention. We will meet again in next episode. Big please make a subscribe on this video so that you will get a further update regarding the new